Okay, here's what should be our final video on the muscle physiology chapter, chapter 9. We're starting at describe 3 ways in which ATP is regenerated during skeletal muscle contraction. This table from your book is a summary of everything that we're essentially about to talk about. So your 3 methods, we've got creatine phosphate, we have anaerobic metabolism, aka fermentation, and then we have cellular respiration or aerobic respiration. So your basics for each one of these that you need to get down. Creatine phosphate is a creatine molecule with a phosphate attached. Whenever you spend an ATP, you release an ADP, which is an adenosine diphosphate. If you add a phosphate back to that, you get ATP. So during creatine phosphate direct phosphorylation, what happens is we take the phosphate off the creatine phosphate and we add it to ADP to make ATP. The byproduct that gets created with this is creatine, which your body is going to deal with. So this gets you a 1-to-1 ratio. You spend 1 creatine phosphate, you get 1 ATP. This can provide most of your muscles with about 15 seconds worth of activity. You don't need any oxygen, you don't need any glucose in order to do this one. Your second option is the lactic acid fermentation, aka glycolysis, aka anaerobic respiration. What happens here is we start the process of breaking down glucose through a process called glycolysis. At the end of glycolysis, you end up with pyruvic acid, and what we do here is we unload electrons from electron carriers back onto pyruvic acid, and that process creates lactic acid. That lactic acid builds up in muscles just a little bit, but it gets released to the bloodstream. The liver will convert it back to pyruvic acid eventually, but you are going to have to deal with this lactic acid. You do not need oxygen in order to do this process. But you do need this glucose that got us started. We make a little bit more ATP than we made during creatine phosphate direct phosphorylation, but not really all that much more. So it's a little bit better than this. You can do a little bit more activity. So your book says 30 to 60 seconds worth of activity can use fermentation pathways. Okay, next, aerobic respiration, or the aerobic mechanism, or cellular respiration. It has a bazillion different names. Starts out with the exact same process. You start this with glycolysis, only instead of using pyruvic acid as a terminal electron acceptor, we keep breaking down pyruvic acid in the mitochondria through 2 more sets of reactions called the Krebs cycle and then the electron transport chain. You end up making way more ATP. This is the theoretical maximum. We don't actually make 38 ATP. We make closer to about 20 ATP this way. You need oxygen in order to do this, and you need the glucose in order to do this. Now, you can use some other nutrients to do this process, but we're going to be just talking about the glucose part. So in order for you to do cellular respiration, you need to have glucose, you need to have oxygen. You're going to make way more ATP, but again, this number is inflated. It's actually going to be closer to 20. You make byproducts, carbon dioxide and water. Your kidneys can deal with that water. Your lungs can take care of that carbon dioxide. Different people have different levels of durability when it comes to cellular respiration. Some people can run like a 5K this way. Other people can't. They tire out after just a mile. Some people can run for days using this process. That just kind of depends on how many mitochondria you have per cell and how's your circulatory system at transporting your oxygen. But these are the 3 ways that you can make ATP, which is again required in order for your muscles to be able to let go and then do the power stroke during muscle contraction. Alright, after that I go into a little bit more detail on each one of those. You can pause during each one of these to make sure you get what you need. I said it all, you just want to make sure you have what's written on those slides written down. Alright, aerobic endurance is how long your specific muscle can do aerobic pathways without switching to one of the anaerobic pathways. And again, that's different for every person, and it's different for different muscles within that person. The anaerobic threshold is the period at which a muscle can no longer carry out aerobic pathways and has to switch to the anaerobic pathways, either glycolysis or creatine phosphate. Again, different for everybody, different for all muscles. Muscle fatigue is the inability to continue contracting. This is not the same thing as I'm just tired and I don't feel like it anymore. This is your muscles literally can't even. I don't know if you guys remember this story, but this woman was trying to run the Dallas Marathon, if I remember correctly. She really wanted to finish that race. Her muscles could not do it anymore. And so this is what muscle fatigue looks like after I get done with this ad.
A marathoner comes within feet of the finish line, but she just can't make it.
This is our winner, ladies and gentlemen. This is not normally what you expect to see at the finish of a marathon.
Chandler Self was leading the Dallas Marathon, but was running on empty. When I watched that video,
I won't keep going because you could see what it looked like. The person who was running with her was running as part of a team, so she couldn't win the race as a solo racer, which is probably why she also decided to help her. But that shows you what muscle fatigue looks like. Again, it's not just you're tired, it's your muscles cannot continue to do contraction. You've run out of ATP, your calcium's not being sequestered like it's supposed to, you got a lot of problems. Now, one of the things that can happen during muscle fatigue is contracture. This is your muscles have run out of ATP. And remember what ATP does, it allows the myosin to let go of the actin. So if you run out of ATP, myosin can't let go, your muscle contracts and then cannot relax afterwards. So again, most people call this a cramp, but in A&P terms, it's contracture. This is also different from contracture that you see in people who have limited mobility. Like if you have a person with nerve paralysis and they can't move their hand anymore, their fingers will start to curve inward because of a different type of contracture. Here we're talking about cramp. Oxygen deficit. So whenever you have been working out, your myoglobin unloads its oxygen. You use the glycogen that had been stored in your glycosomes. You do all of these different processes and you have to rebuild those at the end. The amount of oxygen that you need at the end of your workout to build those stores back up, that is your oxygen deficit. One of the weird things about skeletal muscle goes back to the arrangement of the myofilaments. Because of this arrangement, what it means is skeletal muscle can contract the best when you have it stretched up to about 120% of its total resting length. If you stretch it past that point, like at the extreme end, when you stretch it this much, the myosin can't grab onto actin anymore because it's not nearby, and so your muscles cannot generate tension at this point. So this is called the length-tension relationship. It can just generate the best tension when it's at this resting length right here. Functional characteristics. So there's two things that are going to matter here. It's how fast do the enzymes work that allow us to break down ATP, and it's what do the muscles use in order to generate the ATP. So these are your two functional characteristics that are going to help us classify different muscle cells differently. Now, this is going to incorporate into three different fiber types. Slow oxidative fibers. The slow means it has slow enzymes, and so the myosin can't break ATP super quickly. Oxidative means we're relying on aerobic pathways, so we must have oxygen so that we can generate ATP. The beauty to these muscles is they do not tire out, so they can continue to contract for extended periods of time. Now, earlier I had alluded to dark meat versus white meat. I think the picture was in turkey, but the same thing basically works for chickens. Chickens can run for a long time. Trust me, if you've never been chased by one, they can chase you for a very long time. And if you think about what their drumstick, which is their running muscle, is, they've got a lot of dark meat there. So the drumstick consists of slow oxidative fibers because chickens are really good runners. Now, fast glycolytic fibers are the exact opposite. They have fast enzymes, so this muscle can contract and relax fairly quickly. It's glycolytic, which means it's working on anaerobic pathways, and so we're working with the creatine phosphate and the glycolysis or the lactic acid fermentation. These muscles are strong, but they tire out really quickly. So what I'm showing you in this video is some students don't believe me when I say chickens can fly, so I wanted to show you chickens can fly. They just can't fly for very long. The breast muscle in a chicken is the flight muscle. It connects from their sternum, which is differently shaped than ours, to their wings. And it does have enough energy to allow them to take flight, but because they get tired so fast, they can't do it for very long. And so that's why they have white meat. There's less myoglobin in there. Their enzymes are really quick, so they're powerful, but they tire out fast. There is sort of a best of both worlds scenario going on, and that's fast oxidative fibers. They've got the fast enzymes, so we can do a nice, fast, powerful contraction, but they are oxidative, so they tire out a little bit more slowly than the fast glycolytic from just a second ago. All right, effective exercise on muscles. This is just an FYI. It's kind of like bones. The more you stress them, the stronger they will get. If you stop using muscle for any reason, your body's not going to waste energy making a muscle strong if you don't need it to be strong. And so if you've ever seen a person who had a cast on and they get that cast taken off, you will see atrophy because they couldn't use that muscle while they were in that cast or traction for that matter. One of the quirks of skeletal muscle, and this is something that is debated, is that skeletal muscle can't do mitosis anymore. And so you can't really create more muscle cells through the act of mitosis. So you can't do hyperplasia, which is a term we're going to define in the notes later on. However, there are people out there that have heard the term ripping. Ripping means that you have contracted a skeletal muscle cell so much that you actually tore the sarcolemma or the plasma membrane. That means you damage the cell, but as that cell is repairing itself, it might turn what had been one cell into two cells. This is called hypertrophy. The cell gets bigger and rips. So it's not the same thing as mitosis and cytokinesis, but you can add more muscle. You just have to damage that muscle first by doing the whole ripping thing. Muscles do work in antagonistic pairs, so whatever you can do with like the biceps, you need to be able to do the same thing with the triceps, or you won't be able to reach around towards the back. So notice how these dudes have difficulty reaching towards their center, and it's because they are muscle bound, so you've got to make sure you work your muscles in antagonistic pairs. From there, we start talking very briefly about smooth muscle. So it's got some similarities and some differences. We're still going to have actin and myosin, but we don't get sarcomeres. We don't get all the same connective tissue sheaths either. So which one's present? Only the endomysium. We don't have a paramysium. We don't have an epimysium. Which two layers of smooth muscle are typically present around a hollow organ? There's a longitudinal layer that runs down the length of the muscle, and then there's a circular layer that runs around the organ like the intestines. Peristalsis is a rhythmic wave of contractions where you alternate. You contract the longitudinal layer, then the circular layer. And so you can see peristalsis happening right here as we contract the circular layer, and that would be pushing food through this organ. Varicosities. So back in skeletal muscle, the axon terminal, which is at the very end of the axon, was the only part that could actually communicate with smooth muscle, or pardon me, skeletal muscle. But here we get these little swellings along an axon that are called varicosities, and each varicosity is capable of dumping neurotransmitters onto the smooth muscle. This allows us to contract the entire sheet instead of just a couple of twitchy cells inside of there. So varicosities are just these swellings that are all along the axon that allow us to stimulate multiple places and not just one. Now, back in skeletal muscle, we had T-tubules. Smooth muscle cells aren't as big, and so we don't need T-tubules to go deep into the cell. Instead, we just get these little bitty dips in the plasma membrane or the sarcolemma. That's what caveoli are. Then, our regulatory mechanism is entirely different in smooth muscle. We do not have troponin and tropomyosin. Instead, we have calmodulin and kinase. Also, because we don't have as much SR in smooth muscle, a lot of our calcium's coming from outside the cell, not from the SR. We will still have some coming from in here, but most of it's going to be coming from outside. Calcium is still the key for smooth muscle contraction, but instead of binding to troponin, calcium binds to calmodulin. Now, calmodulin is normally inside the cell and it's inactive. When calcium binds to it, it activates it. Once calmodulin is activated, it activates a kinase, and then kinase is what allows myosin to grab and ratchet to generate the tension. So it's still a multi-step process. It's just that we have a couple of different players. Instead of troponin and tropomyosin, we have calmodulin and then kinase. But we still create cross bridges. We still need ATP. We still need calcium. We just have a couple of those different things. So calmodulin is what the calcium is going to bind to. Once calmodulin is activated, it's going to activate kinase. Kinase is what's going to actually activate the myosin head so that we can form the cross bridge, and then calcium is still that final trigger. Other stuff that's different. Remember in skeletal muscle, if you stretched it too much, it couldn't contract anymore. That's different in smooth muscle. It does not matter how much you stretch smooth muscle, it's always going to be able to contract. The reason I'm showing you this picture is to hit that point home. This is a uterus. When you are pregnant with a baby, it stretches out a lot. If the uterus were made out of skeletal muscle, it wouldn't be able to contract once your baby was this big, but because it's made out of smooth muscle and there's a stretch relaxation response, then we can still contract to push the baby out at the end. Same thing works for the urinary bladder. It doesn't matter how full your urinary bladder gets, your smooth muscle is still going to be able to contract to push the urine out at the end. Unlike skeletal muscle, smooth muscle cells are still mitotic, and so you can add more cells to smooth muscle, so that's called hyperplasia. Single unit smooth muscle must contract as a whole, as one unit. So that peristalsis we were talking about, that is single unit smooth muscle. So this is what you have in the walls of your hollow organs. You do also have a smooth muscle that works a little bit more like skeletal muscle, where it has to be stimulated to contract by the nervous system. It doesn't do its own depolarizations. The classic example I'm going to give you for that is the erector pili muscles. Those muscles, remember, they attach to your hairs and they make your hair stand up when you get cold. If they were single unit, your hair would constantly lift up and lay down and lift up and lay down, but they don't do that. Instead, they contract when your nervous system tells them to, so they behave a little bit more like skeletal muscle in that way. Also, anything that you need a little bit more fine control of, it's going to tend to be multi-unit smooth muscle. So you need very fine regulation to regulate your blood pressure. So the smooth muscle in the walls of your arteries is multi-unit smooth muscle. I already said all that. Myoblasts are stem cells that can create more muscle. Muscular dystrophy is an sex-linked. The most common form of muscular dystrophy is a sex-linked recessive condition, which means it's going to be more commonly found in boys, although girls can get it. It's just rare for that to happen. What happens is, because of a specific protein called dystrophin, their muscles start to atrophy and get replaced by connective tissue, and they gradually get weaker over time. They tend to die fairly young because, remember, your breathing muscles are skeletal muscles, and so they start to fail after a certain point. Treatment for this is going to be along the lines of gene therapy or stem cell therapy, but right now there's only drug trials on that. We don't have confirmed treatments that are FDA approved yet. Sarcopenia is age-related loss of muscle mass. If you work out, you can slow that process or even stop it, but if you don't work out, you will get weaker over time. Atrophy is something that we had talked about earlier. So this is what I was talking about. This person had a cast on their right leg, and notice they've lost a lot of muscle mass on that side. Table 9.3 is a really good one to study the differences between the different kinds of muscle. So you got skeletal, cardiac, and smooth muscle. This one talks about the connective tissue sheaths and how smooth muscle only has the endomysium. This one shows you the triads, terminal cisterna, T-tubule, terminal cisterna. We don't have that in smooth muscle. We also don't get the bands because we don't have the same myofilament arrangement off over there. Now, we weren't talking about cardiac muscle in relation to this. We'll do that more in AMP2. But if you just want to see how it compares to skeletal and smooth, you can. This one talks about axon terminals versus caveoli, motor unit versus the whole thing contracts as one. Here we have our regulatory proteins, troponin, tropomyosin over the actin. Here instead we've got Calmodulin. They left the kinase out of the picture, but that would be what activated the myosin head. So that completes muscles.